With this method, we can study the effects of intrinsic activity on the maturation of cortical interneuron precursors. This technique provides an efficient way for targeting and manipulating the cellular properties of cortical interneuron progenitors that is accessible to the majority of the scientific community.
This protocol involves the meticulous handling of samples and equipment, and it can best be appreciated by visual demonstration.
Using a waterproof pen, draw a straight line across the middle of the bottom part of the bottom surface of 6 35mm Petri dishes. Add 10 milliliters of liquid 4% low-gelling agarose in PBS into 2 of the 35mm Petri dishes and embed 3 to 4 dissected brains in the agarose with the olfactory bulbs facing down per dish across the straight line, leaving 3 to 5 millimeters of space between each sample. When all of the brains have been embedded, place the dishes at 4 degrees Celsius to allow the agarose to solidify, and carve the 3 brains into a single block of agarose, leaving approximately 3 millimeters of gel around the edges of the samples. Glue the block on the surface of a microtome base, and use a surgical blade to cut all the way through the bottom of the block between each tissue sample to obtain 3 independent blocks. Next, use a vibrating blade microtome to section the blocks in ice-cold Krebs solution into 250 micrometer thick slices, using a bent flat microspatula to collect only the slices containing the medium.